Okay, so we are going to go on to what the primary structure of nucleic acids are. So we've talked about nucleotides and nucleosides. Remember, nucleic acids are the polymers of nucleotides. So if you remember, we talked about primary structure of proteins last chapter. That's our sequence of amino acids that's responsible for a lot of the structure. In nucleic acids, the primary structure is the same thing. It's the sequence of your nucleotides. So how we do this is we're going to have the 3' sugar of one nucleotide next to the 5' carbon of the next. So that's what this over here is in blue. The 3' of the first sugar is going to do a reaction, removing water with one of the O minuses of the phosphate. So that's what this bond, oh, not that one. Yes, that one. This bond here is formed in this reaction. And that's how you have the linkage. So this is called a phosphodiester linkage. So we talked about phosphoesters before. This is a phosphodiester because it's on either side. We talked about those a little bit in lipids. And then additionally, we always are going to write our sequence from 5' to 3'. So the first sugar is going to be 5', meaning it's free on the 5' phosphate. And then we'll go down the chain. And then at the very end, we have a free 3' hydroxyl, and that's where we end. So 5' to 3', that's what we mean. Free 5' phosphate to free 3' hydroxyl. So this is a little bit different from proteins. In proteins, we had our amino acid sequence. Nucleic acids, we have our nucleotide sequence. Proteins, we go from the N terminus to the C terminus. Nucleic acids, now we're going from 5' to 3'. And then our bond that connects these for proteins is peptide bonds. Nucleic acids is going to be our phosphodiester bond. So let's do an example of how we put these two together. So draw the condensed structural formula for the dinucleotide CG in DNA. How does the structure differ from the dinucleotide in RNA? Okay. Oh, I even drew this wrong. Okay. So I went ahead and cheated a little bit because these structures take a while to draw. And I just drew out the nucleotides of C and G. So C is the first one in the order that we're given in the sequence. So I'm going to move this little letter over to the side for each of them so that we still remember which ones they are. But our C is going to be first. I'm going to try and move it up just a little bit. Our 5' side, remember our 5' on phosphate is free, so that's where we're going to start. That is going to connect to our 3' position on the sugar. Our G next nucleotide is going to connect to the 3' position. So I'm going to move our other phosphate down, get it ready and in position. I'm going to overlap those oxygens because that is actually what's binding. Let's erase some of this guy here. Let's do blue, why not? So that's our new phosphodiester bond between the 5' phosphoester or 5' phosphate to the 3' of the sugar of the prior one. And then we have our 3' at the end here, and it is a free 3' hydroxide group or hydroxyl group. Okay, so this would be our answer for the dinucleotide CG in DNA. So the reason I said I was wrong to start with is because I had oxygens at these positions. So if this molecule were RNA, these two positions would have an OH instead of an H. Okay, so you guys have practice one here. I'm trying to hide it a little bit because I did have the monomers on there for you. So take a moment, do the same thing, but now do it with a T as your structure. And I'm going to go ahead and scroll down now. So don't look unless you've already done it. But I'm going to scroll down now and do it with you guys. So I drew A and T here. Again, I did them wrong because they are the wrong, they are RNA, not DNA. So let's fix that real quick. Make sure we have the correct one. Okay, so now we have AT. We start with our free 5' position. Our 3' position is going to interact with the 5' position of the next sugar or the next nucleotide. And then we'll have a free 3' position. So again, I'm going to move this guy so that the oxygens are overlapping. And then I'm going to erase said oxygens and draw a new one in. That looks really weird. Let's draw a new one in so that we have our phosphodiester linkage. Okay, so this would be our answer. And then again, the difference, RNA has OH at 2' instead. So we're changing that sugar when we're doing our RNA. Okay, so those are how we make primary structures of DNA and RNA. So remember that it doesn't necessarily have to be just 2. We can do longer and longer and longer. We just started simple.